Hello everyone, welcome back to the last video in this Chapter 2 series for human physiology. In this video, we are going to focus on protein structure and function. We've actually already talked a little bit about proteins back when we talked about enzymes, so a lot of what we're going to talk about in this video, we're going to go back and talk a little bit more about enzymes to put some of that back into the proper perspective. You'll recall that proteins are one of the four major macromolecules that are inside of cells. The other three are going to be kind of an on-your-own assignment for that. Proteins are just so very important in all human physiological processes that I wanted to make sure we had a good, proper video to address that. So let's go ahead and get into this. Now, the first thing to remember is what we talked about in the previous video about how macromolecules are put together. So all of your macromolecules are a polymer of some kind, a polymer that is put together by attaching together individual monomers. Now, for example, carbohydrates and nucleic acids, they are both polymers, but what sets them apart is the identity of the monomer that is used to construct them. Carbohydrates are made by attaching monosaccharides together, and nucleic acids are constructed by attaching nucleotides together. Now, the same thing applies for proteins, except again, in this case, the monomer is going to be different. These monomers are called amino acids. But ultimately, everything else should still apply. These monomers are going to be attached to one another by enzyme-catalyzed dehydration synthesis. Now, when you get to Chapter 3 next time, you're going to learn that in the case of making proteins, this enzyme activity for making proteins comes from the ribosome. So you'll learn all about that in Chapter 3. Okay, so let's take a look at what sticking two amino acids together actually looks like. As with anything else that we've already looked at, you can see that when you get two amino acids close together, and we'll talk about these structures on the next slide, but when you get these two amino acids together, you can actually see that there is an element of water that can be taken away by the dehydration synthesis reaction. So if we lose the element of water, we end up with the formation of a covalent bond in between the two monomers. In the case of making proteins and attaching amino acids to one another, this covalent bond actually has a very particular name. We call this a peptide bond. And it is particularly important because this peptide bond actually ends up being about 1.5 times stronger than your usual single covalent bond. So it's especially tough to break. All right, so let's consider one individual amino acid. A single amino acid is going to have four major parts or four major components to it. Number one, it has what is called the alpha carbon. This is basically the center or nucleus of the amino acid. It's the central attachment point for all the functional groups that are going to make up that amino acid and make it what it is. Attached to the alpha carbon, you have what's called an alpha amino group, or just alpha amine group. This NH2 right here, you'll see is attached to the alpha carbon right here. So when you're forming a peptide bond, that alpha amine is going to react with the next functional group we're going to see here, which is the alpha carboxyl group of the other side of the amino acid. So for actually constructing proteins by sticking amino acids together, number 2 and number 3 in this list are the most important functional aspects of how amino acids can get stuck together. When you are forming a peptide bond, the alpha amine reacts with the alpha carboxyl of different amino acids. So you can see the OH here with the carboxyl and the H here with the alpha amine. That's your element of water that will be lost when you are forming a peptide bond. So if we go back and look at that again, when we're sticking these two amino acids together, we have the carboxyl group of one amino acid over here and the amino group of another amino acid over here. And in case you're wondering, no, the alpha amine and alpha carboxyl of the same amino acid cannot react together. There's not enough flexibility or length in the carbon skeleton of that one amino acid. Different amino acids react together. You can't get the same amino acid to react with itself. And then the fourth and ultimately most important part of the amino acid is what is called the side chain, or R group. So the first thing to understand about this is the R here, that does not stand for any particular chemical element, not the way H stands for hydrogen or N stands for nitrogen, right? So R is not something that you're going to find on the periodic table of the elements. So R is not referring to any one thing there. R actually stands for the remainder of the molecule. So the idea here is that there are a total of 20 different kinds of amino acids that can be used to construct proteins. Now, what makes one amino acid different from the other 19 amino acids? Well, it is not the first three things on this list. Every amino acid is going to have an alpha carbon, an alpha amine, and an alpha carboxyl group. That is set in stone. That is not going to change from one amino acid to another. Now, what does make one amino acid different from the other 19 is the identity of the side chain. So this is actually going to be a different functional group or different atom or sets of atoms between the 20 different amino acids. And that is what is going to make one amino acid unique when compared with the other 19. And like I said, the R does not refer to any one atom. It's just the remainder of the molecule, and that is the amino acid side chain. So the idea here is that because we have 20 different kinds of amino acids to work with, and they all have their own unique side chains, we can actually make a ton of different kinds of proteins depending on things like how long is the protein going to be, meaning how many amino acids are in the chain. It can be infinite for all we care. And then also, what combination of amino acids are we using? So you could make 100 different proteins that all contain exactly 100 amino acids, but if you're using random amino acids, none of those 100 proteins are going to be exactly the same as one another. So there's enough diversity in proteins that you're not just randomly going to make the same protein over and over again, if that's what you're doing. So here's why the side chain is so important. The side chain, because it's made up of different functional groups, and we'll take a look at what some of those side chains look like here pretty soon. When you synthesize a protein inside the cell, you're going to have this long stretch of amino acids with all these different side chains sticking out. Well, depending on which amino acids you use to make the protein and which side chains are sticking out, that whole protein is going to have different chemical characteristics and different propensities to react with other functional groups based on which side chains are there. So one protein may be very apt to participate in this process because it's got these functional groups and these side chains, whereas this other protein over here may not because it's got a completely different amino acid sequence and it doesn't have the right side chains to do what that other protein was doing. So this is why the side chains are so important. They give a protein its chemical and structural and functional characteristics, as we are going to find out here pretty soon. So the first thing that we are wanting to do here is classify these 20 amino acids based on their side chain properties. Even though there are 20 different amino acids, some of them are very similar to one another, and we can just kind of lump them all into the same category, and right or wrong, we can just kind of say that they're basically kind of the same thing. Okay, so these are the 5 different categories as I see them. First, we have nonpolar aliphatic amino acids. The word aliphatic basically just means it's made up of hydrocarbon chains, lots of carbons and lots of hydrogens. Because of that, because this is nonpolar, we call these hydrophobic amino acids. These are not the only hydrophobic amino acids, but they are these themselves definitely are hydrophobic. Then you have polar uncharged amino acids. Typically, these are either going to contain hydroxyl groups or amide functional groups. We didn't talk about amide functional groups last time, but those would fall into the polar uncharged group. And because of their polarity, these would be hydrophilic side chains. Then you have your acidic and basic side chains. They are both going to be hydrophilic because they will either have a negative charge if they are acidic or a positive charge if they are basic, making those side chains very able to interact with water, so they themselves are hydrophilic. And then finally, you have the aromatic amino acids. And chemically speaking, it does have a little something to do with what they smell like, but that's not strictly speaking, why we call them aromatic amino acids. Most important thing to understand about them is that they are also hydrophobic. Now, we're going to talk about each of these side chains, but as we go through them, the main thing I want you to focus on, the most important thing is, are they hydrophobic? Are they hydrophilic? As long as you can frame these side chains within that kind of categorization, you should be fine. Everything else we can kind of not worry about too much. Okay, so here is a list of all of the amino acid side chains, which are shaded in pink here, that are nonpolar and aliphatic. So lots of carbons and lots of hydrogens here. So we have alanine, valine, isoleucine, leucine, methionine, which actually has a sulfur atom here, phenylalanine, tyrosine, and tryptophan. So these are all of your nonpolar hydrophobic amino acids right here. The first 1, 2, 3, 4, 5 of them are aliphatic, and the last 3 of them are aromatic. So the most important thing here, like I said, is that these are all hydrophobic side chains. So if you have a protein that's got a whole bunch of these hydrophobic amino acid side chains sticking out, that protein is probably not going to dissolve in water very well, because we already understand that water does not interact well with hydrophobic nonpolar substances. Now, one thing you may notice about these different amino acids is that they can be written in 3 different ways, either with their full name, a 3-letter abbreviation, or a single-letter code. Regardless, you do not need to memorize any of the amino acids. You don't need to learn the codes, you don't need to learn the abbreviations, you don't need to learn the structures. Come exam time, if I need to ask you a question about a particular amino acid, I'll tell you which category it falls into. I want you focusing more on the categories than the individual amino acids themselves. Okay, so here are the polar non-charged amino acid side chains. You've got serine and threonine, which have hydroxyl functional groups, which we know by now are very, very polar. And then you've also got asparagine and glutamine, which have amide functional groups. So you can see that you've got this CONH2 group. Unlike the primary amine, this does not take on an electrical charge at a neutral pH, but it is still plenty polar, which is why we throw it into this category right here. So these side chains are definitely going to interact favorably with water. So if you've got a protein with a whole bunch of these polar hydrophilic amino acid side chains sticking out, then your protein's probably going to be pretty soluble in water, correct? Like I said, these 4 amino acids you see here, they are hydrophilic because of polarity, but they do not have full electrical charges. Now, there is a 5th amino acid we could mention here, which is tyrosine. Tyrosine was listed on the previous slide. Tyrosine does have a hydroxyl functional group, but still we consider it to be mostly nonpolar. Okay, and then we've got the basic and acidic functional groups. Lysine, arginine, and histidine are the basic side chains. As such, because they're basic, they will take on a hydrogen atom, which is the definition of what a base does. It takes up hydrogen atoms. So they will become positively charged at a neutral pH. And then, so yeah, like I said, that includes lysine, arginine, and histidine. So physiological pH is kind of in the 7 neutral range. Blood is 7.4. Intracellular fluid is 6.8. So these side chains you can assume are going to be positively charged under most physiological conditions. Similarly, with the acidic side chains, aspartate and glutamate, they are acidic, so they give up their hydrogen atoms and they become negatively charged at those neutral pHs as well. So assuming physiological pH of most of our bodily fluids, we can assume that any proteins in those fluids, these side chains are going to be positively or negatively charged in most cases. So what does that get you thinking of? Positives and negatives. Do you recall going over ionic bonding on your own work? Well, what you saw there is that there are electrical attractions between positive and negative charges. That's how you end up with table salts. Sodium, which is positive, and chloride, which is negative, form a strong attraction to one another, which causes the formation of the ionic bond. Well, the same thing happens within a protein molecule. If you've got a whole bunch of these positive charges and negative charges, they like to attach themselves together via what are called salt bridges, which is just a special kind of ionic bond. It's an electrical attraction between the positively charged basic side chains and the negatively charged acidic side chains. And then additionally, we will eventually see that these side chains have the potential to act as effective buffers in biological systems. Acids and bases they may be, but they are actually weak acids and weak bases. And you'll recall that weak acids and bases make for the most effective buffers. So having a whole bunch of proteins like this in your blood actually makes for a very good way of making sure that the pH of your blood does not change too much. Okay, and then here are the aromatic amino acids. So that's phenylalanine, tyrosine, and tryptophan. The only thing that you should really take note of here is how big these side chains are. So the thing that makes them aromatic is the fact that they've got these kind of interconnected ring structures. So phenylalanine, tyrosine, and tryptophan, you can see these interclosed ring structures here. There's a lot of stuff that we could talk about how chemically unique these structures are. The only thing I really want you to take note of, other than how hydrophobic they are, is how big they are. So when we get to the point where we talk about protein folding, having these big cumbersome side chains, trying to fit them into tight spaces ends up being kind of an issue. So that's something to consider when you're talking about getting a protein to fold the correct way. Okay, speaking of protein structure and protein folding, let's do a quick little activity here. Now, first question, how many letters are in the English alphabet? Now, if you said 26, that is correct. There are 26 letters in the English alphabet, and the way that we put words together is that we take any of those 26 letters and we stick them in a variety of different orders and combinations. We have short words. We have some words that are only 2 letters long. We've got some words that are 20 letters long. They can have any number of different combinations of letters in any different order, right? So that means that there should be a nearly infinite list of different words because we have nearly an infinite different number of ways to put those letters together to make the words. So what we're going to see here in this little activity is that building proteins is kind of like just writing really, really, really long words, just with 20 letters or amino acids instead of 26. So we've got 20 amino acids, and we are going to make any variety of different number combinations of proteins, right? We can start with alanine, and then we can go on to valine, and then glutamate. So we can use any of those 20 amino acids. We can make the protein as long or as short as we want to, and we can put any of those amino acids in any different order or combination that we want to. So really, the world is our oyster in terms of how many different kinds of proteins we can make. So just to kind of give you some perspective here, let's say that I want to build a protein that only contains a total of 9 amino acids, right? So that's actually a really, really short protein. We wouldn't even call that a protein. We would actually call that a polypeptide. It's not really long enough to be considered a protein, but we'll just go ahead and roll with it here. So let's say I want to build a protein that contains exactly 9 amino acids. There are really no restrictions here. It can contain any of the 20 amino acids in any order. Any amino acid can be used repeatedly, so this could literally just be 9 alanines stuck together or 9 glycines stuck together, or we can omit any of the amino acids, and we're obviously going to have to if we only have 9 amino acids. There's no restrictions here. I can put any of the 20 amino acids together in any combination I want. The question is, how many different kinds of proteins can I build with that lack of restrictions? So if I went through every single different possible combination of proteins that are 9 amino acids long, how many proteins would there be? Well, the answer here is that we could build 20 to the 9th power different combinations of proteins here. 20 is the base because that's the number of amino acids I have to work with. 9 is the exponent because that is how many amino acids I want in the protein. If you do the math there, that is 512 billion different combinations of proteins. 512 billion different combinations of proteins that contain a total of 9 amino acids. And let me blow your mind even more. Most human proteins are well over 100 amino acids long, so that would make the number exponentially greater than 512 billion. If you tried to do the math in that case, your calculator would start giving off smoke and it would explode, right? So there are, by all estimates, about 30,000 different proteins in humans, but we just established that there are billions and trillions and quadrillions of different possible combinations of proteins that we could possibly make. So with all those different possible combinations that we could make, why do we only have 30,000 of them? It's an interesting thing to think about, so we'll kind of talk about that a little bit later on. All these different possible combinations of proteins, but yet we only use a very teeny tiny percentage of those possibilities. Okay, so let's talk about what you do with a protein once you have all the amino acids chained together. So once you have that kind of long linear chain of amino acids, you can kind of think of a string of pearls. That is what you would call the primary structure of the protein, right? So you're literally just talking about what is the amino acid sequence? How long is the amino acid? How long is the protein? How many amino acids does it contain? And which amino acids go in which order? That is the primary structure of the protein. But the problem here is that if that's all you do with the protein is not going to have any sort of physiological or cellular function, because what we're really going to hammer home here is the fact that proteins cannot function if they do not have three-dimensional shape. Shape is by far the most important aspect to how a protein does what it is supposed to do. The protein has to fold into a very specific three-dimensional shape in order for it to attain its intended physiological function. So because the 3D structure of a folded protein is ultimately going to be determined by the primary structure, so the primary structure has all the underlying information we need to fold the protein correctly, we just need to actually go through the process of folding it in order to get it to do the right thing. But it cuts two different ways, right? So the downside to that is that if the primary structure is all the information you need in order to fold the protein correctly, if we start swapping out amino acids and changing the primary structure, we're going to change the way the protein folds. So if we change the way the protein folds, it's possible it's not going to work correctly anymore. And we'll have something to say about that a little bit later. It's actually something that happens in genetic diseases. So if we put this all into perspective, the fact that when a protein in its primary structural configuration folds, it has to have a very, very specific three-dimensional structure. Most three-dimensional structures do not lead to functional protein, and that helps to explain why out of the orders of magnitudes of trillions and billions and quadrillions of different possible combinations of proteins, only a very small number of them actually fold into a structure that is useful to human cells. Hence the 30,000 total human proteins compared to all those trillions of different possible combinations. All those other combinations that aren't a part of the 30,000, they must not either fold correctly or they don't fold into any sort of useful thing for our cells. They don't have any physiological function. Okay, so if you actually look at this figure here, you can actually see the step-by-step process of how we get a primary structure protein to fold into its three-dimensional shape. We go through an ordered series of folding steps going from primary to secondary structure, you can see right here, secondary to tertiary structure, and then finally tertiary to quaternary structure. So let's start with secondary structures first. Secondary structures are formed as the result of amino acids within the protein that are in close proximity to one another, meaning they are less than 10 amino acids apart from one another within the primary structure. They start interacting with one another, and this is actually going to require the carbon skeleton of the protein to actually have a little bit of flexibility with it to allow those amino acids to come together. So there are two different types of primary structure. These are called alpha helices and beta sheets, or beta strands. Now, if you look at what we're looking at here, the alpha helix, true to its name, kind of takes on this ascending staircase structure in which you get these stabilizing forces that hold the part of the staircase together between an electronegative oxygen and a partially positive hydrogen. Do you remember what that kind of bond is called? Because we also see it here in the beta sheet. You see these interactions between an electronegative oxygen and a partially positive hydrogen. Well, if you said hydrogen bonding, that is absolutely correct. Hydrogen bonding is the major force behind the formation of secondary structures. So hydrogen bonding isn't just for water, and it's not just for DNA molecules. Hydrogen bonding is very important for holding proteins together and getting them to fold correctly. So these bonds are actually formed specifically between the atoms of the polypeptide backbone. If you look at the R groups here, you can see that the R groups really are not participating in any of these stabilizing hydrogen bonds. The R groups will come into focus next time when we start talking about tertiary structures, but for secondary structures, hydrogen bonding is the name of the game. So our secondary structures here are the alpha helices, in which the carbon skeleton kind of winds around itself like a staircase, and that structure is held together by hydrogen bonds that kind of lock it into a configuration and prevent it from coming apart. And then finally beta strands, in which you kind of bend the backbone back across itself in either a parallel or anti-parallel fashion, and then oxygens and hydrogens kind of latch onto each other from either side of the turn. Okay, so once you've formed a certain number of secondary structures, we can move on to tertiary structures. Tertiary structure is achieved by interacting amino acids together that are far away from each other. Now, what we're going to see here is that if the amino acids are far away from each other in the primary sequence, that means that it's going to have to be the side chains that are interacting rather than the backbone. And we've already had a little discussion about the different kinds of amino acid side chains and what their chemical reactivities tend to be. So tertiary structure is going to result from more than just hydrogen bonds. Now, hydrogen bonds can happen here, but it's not just hydrogen bonds, not the way it was with secondary structure. Secondary structure was just hydrogen bonds. With tertiary structure, we can get hydrogen bonds from different side chains, so this might be a serine and this might be a carboxylic acid. We can have hydrophobic interactions in which we get two hydrophobic side chains interacting together just because they're both wanting to get away from water. You can get ionic bonding. That's what we talked about with salt bridges and the acidic and basic side chains that we talked about before. And then finally, you can get another type of bond that we didn't talk about before, which is called a disulfide bridge, a special type of covalent bond that is forged between two different cysteine side chains that both contain sulfur. So once you get your secondary structures, and then you can kind of start twisting the backbone around to get some of these side chains to interact with one another, you're going to have twisted the protein around so much that it's actually going to start taking on a discrete three-dimensional shape, as you can see here in the case of something like myoglobin, which is what this is supposed to be down here in the bottom right. Now, for a single polypeptide, a single continuous polypeptide like myoglobin, the tertiary structure is going to be as far as you go with the folding process. That is the final three-dimensional shape of that particular protein because that protein exists all by itself. So we're not actually going to take it any further than that as long as we are talking about one individual polypeptide that acts all by itself. So we'll have more to say about cases in which that doesn't apply a little bit later on. So let's talk a little bit more about disulfide bonds. Disulfide bonds, just like any of the other tertiary structures, it's there to help lock the protein into its folded configuration, except because this is a covalent bond, it is a little bit more permanent. It's not strictly permanent, but it locks you into place in a way that hydrogen bonds and ionic bonds just cannot do. This is especially helpful for proteins that are exported out of the cell, and they get away from the machinery inside the cell that could refold them if they happen to unfold themselves. So disulfide bonds are all about the amino acid cysteine. It's the only amino acid that has this particular functional group, which is called sulfhydryl. It's like a hydroxyl group, except with a sulfur instead of an oxygen. So if a protein is going to have any disulfide bonds, that protein obviously has to have at least 2 cysteine side chains in the primary structure. So 2 cysteine side chains interact together to form one disulfide bond. Okay, let's do a quick little activity here just to make sure that we understand how amino acid side chains like to interact together. So you can pause the video at this point and then you can replay it so that you can see the answers here. But the idea here is that you are going to match up the amino acids on the left side of the screen with the amino acids on the right side of the screen to come up with side chain tertiary structure interactions that make the most sense. So all you have to do is draw a line from, say, alanine to one of these 4 over here to come up with the interaction that makes the most sense. So I'll give you a second to pause the video, and then when you come back, we'll reveal the answer. Okay, so if we start with alanine, alanine is a non-polar side chain. So the question is, which one of these 4 over here is it going to want to interact with? Cysteine, which has a free sulfhydryl, threonine, which is polar and has a hydroxyl functional group, glutamate, which is acidic, or isoleucine, which is also non-polar. Well, if you recall something important we said in a previous video, like goes with like, polar goes with polar, and non-polar goes with non-polar. So this would be an example of a hydrophobic interaction, 2 non-polar side chains huddling together, if for no other reason than to make sure that they get themselves away from water, their arch nemesis. Okay, next let's look at arginine. That is a basic amino acid side chain, so it's going to be positively charged at a neutral pH. So which of these remaining 3 over here is it going to want to interact with? The sulfhydryl group, the hydroxyl group, or the acidic group, which is going to be negatively charged at a neutral pH? Well, that is your answer there. Positives and negatives go together. When arginine and glutamate come together as the protein folds, that forms what we call a salt bridge. Next we have cysteine. Well, we just got done discussing that when 2 cysteines come together, they form a disulfide bond. So as the protein folds, we're going to want to get cysteines in close proximity to one another so that they can form their disulfide bridges. And then finally, what that leaves left is serine, a polar amino acid side chain interacting with threonine, another polar side chain interaction. So this would be an interaction that is stabilized by hydrogen bonding in all likelihood. Okay, so let's talk a little bit more about protein folding and protein shape. So we just got done saying that the shape, the 3-dimensional shape of a protein, that is by far the most important aspect of its function. So as long as you can get a protein to fold into the right shape, it should, in all likelihood, function the right way. We're going to see plenty of examples of that as well. But what happens if you have a fully folded protein that suddenly becomes unfolded? The unfolding of a protein is a process called denaturation. Now, let's think about this. You've got the folded protein and it suddenly becomes unfolded. Is that really such a big deal? All the amino acids, all the side chains, chemically, the protein is still all there. All the side chains and all the functional groups are still there. The protein has not been really damaged in any particular way. We haven't changed the chemical nature of the protein. We've only taken away its 3-dimensional structure, so we have denatured it. Now, here's what makes it so important that proteins maintain their 3-dimensional shape. If you denature a protein, even though the protein's all still there, chemically it's all still there, protein's not going to have a lick of function. So the protein is basically dead at that point, and you might as well throw it in the trash and start over. This is why it is so important that proteins fold the correct way, because folding and 3-dimensional shape, that is 100% the story as it pertains to protein function. So if that sounds kind of scary, if you're worried about your proteins denaturing, let's talk about what actually causes proteins to denature. So as long as conditions in the cell and in the human body are favorable for protein folding, you shouldn't have to worry about this too much. But there are some conditions that make it favorable for a protein to become denatured. The most famous example of this is excess temperature. If you expose a protein to high temperature, that is going to break all of the hydrogen bonds that are stabilizing the secondary and tertiary structures. Without those hydrogen bonds, the protein can't stay folded and it falls apart, as we've become aware of by now. Another thing you can do to denature a protein is to change the pH. What that does, you'll recall we said acidic side chains are negatively charged, basic side chains are positively charged, and what we always say with that, the caveat we always give there is that's at neutral pH. If the pH is neutral, acidic side chains are negative, basic side chains are positive. What if the pH isn't neutral anymore? If the pH is no longer neutral, either the acidic side chains are then going to become neutral and no longer negative, or the basic side chains are no longer going to be positive and they're going to become neutral. If we do just one of those two things by mucking around with the pH, you take away the ability of proteins to be stabilized by salt bridges, and guess what's going to happen? Protein's going to fall apart because you are taking away those stabilizing electrostatic interactions. So an example of this would be next time you're at home and you're in your kitchen, if you have a little bit of milk and lemon juice to waste, pour yourself a glass of milk and then add a bunch of lemon juice to it. See what happens to the milk when you add lemon juice, which is a very strong acid. The proteins in the milk are going to curdle, and the reason why that happens is because you have disrupted the way those milk proteins have folded, and they're all going to lose their solubility in water, and they're going to fall to the bottom of the glass. In addition, chemical agents, drugs, and as well as the natural aging process for diseases like Alzheimer's disease, these can cause proteins to unfold and denature as well, but we won't worry about that too much in this regard. As far as you're concerned, temperature and pH changes are the major ways in which denaturation can occur. And here's a good meme that actually kind of illustrates the temperature aspect of it. So when you get sick, if you get, say, a bad cold or a sinus infection, you're probably going to be running a fever, right? So you're running a fever of maybe like 100, 101. Well, the whole idea there is that by increasing the temperature, we can kill off the bacteria or cause the viruses to, the viral proteins to denature and unfold. So if you look at this meme here, you're asking, okay, well, why do I have a fever? Immune system saying the virus can't survive high temperatures, the bacteria can't survive temperatures. Well, why can't the virus and why can't the bacteria survive high temperatures? Well, for the same reason that we can't, because the proteins in the bacteria and in the virus can't stay together. They lose their shape, they lose their structure, they lose their function. But obviously the double-edged sword there is that for us, we don't really like the high temperature either, so it's not really in our best interest to be running a fever for very long, which is why it's important for us to have a hypothalamus to make sure that our temperature doesn't run too high. There is your reference back to chapter 1 and homeostasis. Okay, so a few more things. Let's put the denaturation process in a little bit of extra context. Let's talk a little bit about enzymes again. You'll recall way back when we talked about chemical reactions, we said that as it pertains to temperature, you can only speed up a chemical reaction by cranking up the temperature so much before you get diminishing returns. Well, those diminishing returns come in the form of denaturing enzymes that are there to speed up the chemical reaction. So if your temperature is running too high, the enzymes are going to unfold themselves, and at that point, they are not going to be catalyzing any reactions. They have no function at that point. If you look at the picture here, substrates are meant to fit into a very particularly shaped active site for that enzyme. Again, this is why this substrate fits into this enzyme's active site and not other enzymes' active sites. But with excess temperature, the shape of the active site is drastically going to change such that the substrate doesn't fit there anymore, and that reaction is no longer going to be catalyzed by this enzyme, and you're back to the point where you're dealing with a reaction that is technically spontaneous, but because there's no enzyme, it's going to be very, very, very slow. So this should make you aware of, for any given organism, every organism has what you would call an optimal temperature. This optimal temperature at which the speed and rate of chemical reactions is absolutely at peak efficiency. And this is the temperature, it's no coincidence, this is the temperature at which proteins stay folded together and they are able to catalyze reactions. So for humans, this temperature is 37 degrees Celsius, or 98.6 degrees Fahrenheit. If you've ever wondered why it is that our body temperature is what it is, well, say no more. We want it to be the temperature to be sufficiently high to get molecules to collide with one another at a good rate, but not so high that our proteins start unfolding. And it's worth mentioning that different organisms are each going to have their own optimal temperature. So, for example, some bacteria that we're familiar with, especially those that are in our gut, like E. coli, they also work at an optimum of 37 degrees Celsius. But some bacteria that like to live in like hot springs actually have an optimal temperature more around like 95 degrees Celsius, almost the boiling point of water. So every organism is going to be different. They have evolved in such ways that their proteins can withstand different temperatures in different ways. So it's worth mentioning at least. Okay, and then finally, let's talk a little bit about quaternary structure. This is the fourth level of protein folding, but this is only going to be applicable when you're dealing with a protein that is made up of multiple subunits, meaning that this whole protein consists of multiple different proteins that have kind of interacted together to form one big complex. Now, for something like myoglobin, which is just one single polypeptide, myoglobin does not have a quaternary structure. It's just one polypeptide that exists all by itself. Now, hemoglobin, which is somewhat similar, it's a little bit different. Hemoglobin is not one individual protein. It is actually four different proteins that have conglomerated themselves together into a big complex. Hemoglobin has quaternary structure because it is made up of four different polypeptide subunits. They are called globin proteins. Hemoglobin itself consists of two alpha subunits and two beta subunits. Each of the subunits here, you'll notice, have their own tertiary structure, but when these tertiary structured proteins then come together and bind to each other, that's when we're talking about quaternary structure. Now, in the case of hemoglobin and myoglobin too, we can go ahead and throw it in there as well, an interesting case occurs when a protein actually contains a chemical component that is actually not derived from amino acids. In those cases, we call that a prosthetic group. Kind of like if, say, someone lost a leg or lost an arm in the war, they can actually get themselves a prosthetic limb. It's not something that grew on them. It's not something they've had all the time. It's not necessarily natural, but it actually has some function with respect to the rest of the body. In this case, with globin proteins like the globins in hemoglobin and myoglobin itself, they all have what is called a heme prosthetic group. So this molecule of heme you see over here on the far right, the most important aspect of what's called the porphyrin structure that you see here is that at the center is coordinated a single iron atom. And this gives to the heme prosthetic group the ability to bind to a single molecule of, I'm sure you know where we're going with this, oxygen. So the fact that these globin proteins have this heme prosthetic group, that means that these proteins have the capability of binding to and carrying oxygen. Other proteins that do not have these prosthetic groups cannot bind to oxygen. So prosthetic groups can give a protein a function that it otherwise would not have. Now, real quick, and we're going to learn more about this much later on in the semester when we get to the respiratory system, But let's do a quick little structural comparison of hemoglobin and one of the subunits, or of one of the subunits of hemoglobin and myoglobin. So myoglobin is just a single protein, so this is what it looks like 100% of the time. And then let's compare that to one of the beta subunits of hemoglobin. So we just take one of those beta subunits off of hemoglobin, and we put it right next to myoglobin here. Do you notice anything here? Do you notice how extremely similar they look to one another in terms of their structure and three-dimensional shape? They've got all the same alpha helices. They've got the prosthetic group in exactly the same spot. There are a few minor differences here and there, but for the most part, these look like nearly identical proteins. So you would expect them to have, correct? Well, that's not the case. Look at this graph right here. This is a oxygen saturation curve in which we compare the ability of myoglobin and hemoglobin to bind to oxygen. These graphs are a little bit tricky to read, so let me kind of point your eyes in the right direction here. On the y-axis here, this is a percentage, 0% and 100% at the top. This is telling you of all the protein, whether it be myoglobin or hemoglobin, what percentage of that protein is bound to oxygen. If it's at the top, it's 100%. If it's at the bottom, it's 0%. No oxygen, all oxygen. Got it? Okay, and then on the x-axis, we have got the partial pressure of oxygen in solution. So picture there being a certain amount of oxygen free in your blood. So the idea here is that as the curves go up, you'll notice up here at the top, at very high concentrations of oxygen, we get these proteins fully loaded up with oxygen. So at very high levels of oxygen, about 100% of both myoglobin and hemoglobin are bound to oxygen. Well, where these curves are different is kind of here at the lower concentrations of oxygen. At the lower concentrations of oxygen, let's say 25 torrs of oxygen, you'll notice that hemoglobin is only about 50% saturated, whereas myoglobin is still pretty close to 100% saturated. So clearly there's something about the difference between myoglobin and hemoglobin that causes them to bind to oxygen in very different ways. Now, we're not going to belabor that point here. Like I said, we'll talk about that a lot more in later chapters, but it's just something I want you to think about. What is it about the difference between myoglobin and hemoglobin that causes them to behave so differently in terms of a functional aspect, in terms of how they bind to oxygen? So think about what we've said about the difference between myoglobin and a whole complex of hemoglobin. Okay, so I know this has been kind of a long video at this point, so we have just a few more things to get through and then we'll be done. Let's talk about protein shape. Proteins take on one of two major different shapes. You can either have globular proteins or fibrous proteins, and this is going to be important because this has to do with the way we like to categorize everything in terms of whether it's hydrophilic or hydrophobic. So a globular protein like myoglobin over here is going to take on the kind of rough appearance or rough shape of a sphere. And in doing so, you tend to have your hydrophilic side chains pointed towards the outside to where the fluid is. So globular proteins tend to be very hydrophilic and they tend to be very soluble in water. So you tend to have acidic, basic, and polar side chains exposed to the surface of the protein, and then you've got those hydrophobic side chains kind of buried towards the center. Fibrous proteins do just the opposite. They tend to be very hydrophobic because their hydrophobic side chains are pointed to the outside and their hydrophilic ones are pointed towards the inside. So fibrous proteins like collagen or keratin tend to kind of polymerize themselves into tangles or fibers or rods, and they do not dissolve in water very well. So what do you think? Do you think globular and fibrous proteins are similar or dissimilar in their primary structures? And if they are or if they are not, why would that be? Why would one protein be very hydrophobic and another not be? So we've already seen at this point that most enzymes are proteins, but since we've already talked so much about enzymes and the way that proteins fold, we want to make sure that we completely understand this concept. So almost every enzyme, with the exception of the enzyme activity you see in ribosomes, which comes from RNA, almost every enzyme itself is a fully folded protein. And as we mentioned before, An enzyme is only going to be able to catalyze its particular reaction if it folds correctly, if it's able to engage its particular substrate in the correctly shaped active site, right? So an enzyme, if we talk more about the active site, the enzyme has this active site, which is just kind of a special binding pocket for that particular substrate. It's got a shape that perfectly matches the substrate that's going to come in. So this ensures that the enzyme is only going to catalyze one particular reaction and not a whole bunch of other ones. Enzymes are not promiscuous in terms of the reactions they catalyze. They stick to just one reaction, and that's pretty much it. And then another thing to at least be aware of here is that in the active site, the reason why enzymes can catalyze reactions is that particular amino acid side chains are sticking into the active site, and those side chains can themselves participate in the chemical reaction, whether it's breaking bonds, forming stabilizing intermediates. Ultimately, that is how the enzyme reduces the activation energy and speeds the reaction up. Sometimes just having a fully folded polypeptide for an enzyme is not going to cut it. Sometimes you need other things for activity. Some enzymes bind a metal ion cofactor, like zinc is a cofactor for the enzyme we've talked about before called carbonic anhydrase. You've got to have zinc in order for this reaction to happen. Another one we've talked about, in some cases, enzymes require coenzymes. Usually these are just going to be vitamins like vitamin C. Back in Chapter 1, we discussed how vitamin C is necessary to prevent yourself from getting scurvy, and all we really said is that vitamin C is in some way necessary for making collagen. Well, it's not actually required for making collagen, but it is required for getting the triple helix structure for collagen that is necessary for its stabilization to occur. So once these collagen polypeptides are made, two different enzymes called lysyl hydroxylase and prolyl hydroxylase attach a whole bunch of hydroxyl groups to the polypeptides, and then you get those polypeptides to stick together by a whole bunch of hydrogen bonds. Well, these two enzymes we're mentioning here require vitamin C as a coenzyme. And if you have a vitamin C deficiency, those enzymes are not going to work correctly, collagen's not going to get made properly, and you're going to end up with scurvy. Enzyme activity can actually, from external forces, greatly be slowed down by a group of chemicals called enzyme inhibitors. There are two major types of inhibitors we're going to talk about here. Competitive inhibitors are molecules that very much resemble the substrate and can engage the active site, but they're structured in such a way that the enzyme can't catalyze the reaction. So basically, when the competitive inhibitor is there, the substrate can't get in there and the enzyme can't do its job. So that will slow the enzyme down because you're basically causing it to be busy with something that is totally non-productive. Non-competitive inhibitors do not engage the active site, but rather a different site on the enzyme, which is called an allosteric site. When the non-competitive inhibitor binds, this will actually change the way the enzyme folds, and the active site is not going to look the same anymore, and the substrate will not be able to bind. So those are the two major types of what we would call reversible inhibitors, where the inhibitor can be washed away and you can restore enzyme activity. Irreversible inhibitors actually permanently destroy the enzyme activity and are sometimes considered poison. So if you've ever heard of something is poisonous to you, more than likely it is irreversibly inhibiting one of your cell's enzymes. And the reason why we bring this up is because a lot of pharmaceutical drugs themselves are enzyme inhibitors. So the way this generally works is that if you suspect that one of your patients has a particular disease, and if that disease is characterized by an enzyme maybe not doing what it's supposed to or doing something it's really not supposed to be doing, then you can give an enzyme inhibitor to kind of modulate the activity a little bit to get it back to where it's supposed to be, in all likelihood. So thank you for your patience and your attention. I know that was a lot to go through, but the reason why we had to go through all that is because you are going to come to appreciate in the next chapter and in all of the coming chapters just how important protein function is. So, for example, in the next chapter, when we talk about membrane transport, we're going to talk about the importance of all different kinds of proteins that you can find in the plasma membrane. And proteins aren't just found in the plasma membrane. You find plenty of them in the cytosol and other places too. But you're going to become familiar with how all these different proteins work, like channel and carrier proteins that can transport molecules across the membrane, receptor proteins which mediate cell-to-cell communication, which we'll be talking about pretty soon. Enzymes can be in the cell membrane. Junction proteins can hold cells within tissues together. Cell recognition proteins function in things like blood typing and the immune response. So you're going to come to appreciate that proteins are at the center of every physiological process. So it's really important that we understand at least a fundamental level how proteins are put together and how they generally function. All right, so that is going to do it for this video and this chapter. Thanks again for your attention, and I will see you next time with